Okay, hello everybody. So we are at the top of the brainstem this week, and obviously there'll be midbrain. I just want to say that the last several lectures on the medulla of the pons and midbrain, and first of all, they complete a group organized by brainstem, but they also are a group in the sense that in many ways they are the most difficult lectures of the entire course, because unlike every other topic before and every other topic which will come, they're not focused on any one system or type of activity, because we're just looking at the anatomy of the brainstem and learning it that way. And there's just a lot of things going on and a lot of names that are anatomically based and things to remember. After this, it's going to become much more systems in the central nervous system, and in many ways will get easier to listen to, although there'll be just as much detail. So here's our outline for today. If there's one theme that we will be focusing on, it's the control of eye movements, which are throughout the midbrain, but mostly in the rostral half. So on we go. The transition from the pons to the midbrain. We left pons at the end of the last talk. And so you'll know that you're in the midbrain when the fourth ventricle becomes the aqueduct instead. That's the easiest way to know it. If you see the aqueduct of Sylvius, clearly you're in the midbrain. You'll also see the superior medullary velum disappear and instead replaced with a tectum, which is the first time that we will have had a tectum, a roof of the section of the brainstem in the midbrain. Another way in the caudal portion of the midbrain is the decussation of the superior cerebellar peduncles, and finally just a smaller tegmentum in general as the tectum becomes real. Here's a posterior view of the midbrain, and you can see the colliculi. So the inferior colliculi are how you know that you're in midbrain, or the superior colliculi if you go up higher. You're also going to be seeing the decussation, the superior cerebellar peduncles, and their decussation in the caudal portion of the midbrain. So what do we got going on in there? We have the inferior colliculi. We have another nucleus called the parad bigeminal nucleus, and the trochlear nucleus, and then the tegmental and interpeduncular. Again, it's names like this that drive you crazy because they don't tell you what the tracts are for, and that's really the problem. They're named for where they are. Well, here we are at the transition. We're clearly looking at basis pontus, and yet here's the inferior colliculus. So let's just look for a moment. Here is the inferior colliculus in here, and we're going to look at it again. And you can see the nuclear structure surrounded by a lot of fibers. Here's the commissure of the inferior colliculus here. There's the cerebral aqueduct. Tough to see, but you can sort of see the ependymal lining helps outline this almost diamond shape. And surrounding it is a much larger area of the periaqueductal gray, which we're going to look at later. The MLF is still sort of in our favorite position in the midline posteriorly within the tegmentum. Here's the superior cerebellar peduncle, and here's the decussation of the superior cerebellar peduncle. Medial lemniscus, again, we saw this at the end of the last talk. Medial lemniscus is way out lateral. Here we have the central tegmental tract, which we had talked about as a tract of ascending information coming from the rostral nucleus solitarius, heading up to the thalamus with taste information. The lateral lemniscus, which we will deal with later in the course, but also later in this lecture as a pathway for ascending sound. All right, look at the parabigeminal nucleus. This is the best slide for this. I'll return it later when we talk about it, but it's way out here on the surface of the tegmentum, strictly, of the midbrain. Not a nucleus that we think about a lot, and indeed, it's some of these nuclei that we're going to talk about. They don't play a major role in our clinical thinking because they are part of larger systems, which when the neurologic deficits don't localize easily, is what I would say. All right, let's focus on the inferior colliculus. This is a major center for auditory fibers. That's basically what's going on. And it has three parts. It has a central nucleus, a pericentral nucleus, which is dorsal to it, and an external nucleus. We're going to look at each. The central nucleus is laminated. and it has tonotopic isofrequency contours. And that's basically, it's functioning as a relay nucleus of the incoming sound information heading up to the medial geniculate, and we'll talk about that in a minute. The other two nuclei are not organized this way. They have more to do with attention. The external nucleus is related to the acoustic-motor reflexes for orienting our head and eyes, as well as our ears, towards sound. And the pericentral nucleus doesn't turn your head, but it focuses your attention on a certain part of space through focusing on acoustic information. The inferior colliculus, as we say, is an auditory relay. It transmits impulses up from the lateral lemniscus, bringing things up from the brainstem, and then transmits them to the medial geniculate. The medial geniculate is what sends all of that auditory information up to the auditory cortex. And because of that, as well as its location, the medial geniculate is considered equivalent, at least, and often considered part of the thalamus. All right? It's not in the thalamus structurally, but it's nearby, and it's certainly serving a very analogous function, unlike the superior colliculi, which are not going to be that way. The dorsal medial portion of the inferior colliculus neurons, they have a lot of commissural fibers, so everything is bilateral. And in fact, most of the inferior colliculi neurons, but most of the neurons in the brain are binaural. There's not a lot of, hardly any single ear neurons. And that's because as localization in space, okay, requires binaural input. And so, and it's monosynaptic. It's right from the start in the trapezoid body. And it's really, hearing is never monaural again. All right, and so that's an important principle also in clinical localization. There are no central nervous system causes of unilateral hearing loss. They don't occur, all right? So when you're trying to figure out if something's central or peripheral or unilateral hearing loss puts you out at least on the lateral aspect of the pons or distal to that, never central to that. All right. Here's the inferior colliculus, cresol violet stain, and you can almost make out an ovoid, almost, not really, but on the next cartoon, it's easy to see. It's so much easier when they draw it for you, right? So here's your central nucleus with the isofrequency map. Here's the pericentral nucleus, that is for auditory attention, and here's the external nucleus, that is for acousticomotor orientation, basically so you turn your ears, you can say turn your head, or you can say turn your ears, because the ears are pretty fixed in your head. So when you do one, you do the other. But you orient towards sound and actually execute that as the external nucleus as participates. Looking in more detail now, here is the commissure of the inferior colliculus. Here's the inferior colliculus. Here is the region of the central nucleus. Here's the brachium. Now the brachium, the brachium of the inferior colliculus is efferent. The brachium of the superior colliculus is afferent. Okay, so they're really quite different structures. They look the same, they're next to each other. We call them the quadrigeminal plate as if they're four twins. They're really, really different from one another. They play fundamentally different roles in the visual or auditory system. Well, we looked at that parabigeminal area nucleus. What is it doing out there? So it's ventrolateral, way out on the side, ventrolateral to the inferior colliculus. It's a well-defined nucleus, we saw. These are cholinergic neurons. What are they doing? They receive visuotopic information from the superficial layers of the superior colliculus. So we're talking about them because they're in caudal pons, but we're going to return to them when we get up into the rostral pons and think about superior colliculus. They get their input from the ipsilateral superior colliculus. They project to the ipsilateral and contralateral superior colliculus. What are they doing? They're very important. They are for error detection between eye position and target. All right. They also play a role, and this is known, but mostly physiologic data. If you stimulate these parabigeminals, you get a deer in the headlight reaction. The person is stuck on the vision that they're looking at. They're stuck on it. Okay, so they are important for detection between eye position and target, and yet in abnormal circumstances, they contribute to this freezing phenomena set off by a startled vision coming right at you. Just when when you should, the least time that you should be freezing, you do. They play a role in that. But their main physiologic function is to detect an error, not necessarily to correct the error. Somebody else is going to correct the error. We're going to look at that later when we get into the rostral. And so we might as well start to make this point, and this is why I was saying you're tough for localization, because this is a distributed network. The whole reticular formation, there are going to be little nuclei that we can name, we can identify them, but they're so interconnected that if the person can't follow a target, what do you want to, whose fault is it? What nucleus do you want to attribute it to? And the answer is, I don't know if this is superior colliculus, if this is, the interstitial nucleus of Cajal, if this is the parabigeminal area, I don't know. And that's what I mean when I say it doesn't localize well, except to say that they're all in the midbrain. Okay, in that sense, it localizes very well. All right, so let's keep going here. That's the parabigeminal, and that was out here. And these are all discrete nuclei. You can really see them very well. All right, of course, continuing the theme of oculomotor, in the caudal portion of the midbrain is the trochlear nucleus. It's a small caudal appendage. I really think you can think of it as part of the oculomotor complex, which is a full complex, is a good word. Obviously, it innervates the superior oblique. Its root fibers, as you well know, emerge dorsolaterally, all right, and then and caudally, all right, to in the region of the periaqueductal gray. They then decussate completely, all right, in the superior medullary velum, and they exit ipsilaterally to the superior oblique that they innervate. All right. So here you go, like that. And here from that picture that we saw before, I pulled these out. And this, assuming this is the left, this would be right, since we're looking from behind. This will be the right fourth nerve. It came from the left trochlear nucleus and crossed before it emerged from the brainstem. All of these nuclei are quite discrete and easily recognized. And here's a trochlear nucleus right here. We're also going to be talking about the dorsal tegmental nucleus. We'll come back to that character later. We use the term periaqueductal gray very frequently, and what do we mean? It is a region where the nuclei are actually, in this case, kind of somewhat difficult to see, and they all tend to blend toward one another, although once you start staining for looking at shapes of neurons, they're identifiable. In the periaqueductal gray, we include, first of all, the mesencephalic nucleus of the trigeminal. We've talked about that. We're not going to talk about it anymore. And the locus ceruleus, and we talked about that last week. We talked about some of the raphe nuclei, but we're going to talk a little bit more now today. I mentioned the dorsal tegmental, and then there's this whole midbrain that we just refer to as a periaqueductal gray, and it doesn't get another name. And so here in a sort of sagittal view is the locus ceruleus will be here, and the raphe nuclei are all over the place. All right, and then the periaqueductal gray, and this is a cartoon. But here you are. So here's the mesencephalic. So here's all of this, right? Would be periaqueductal gray, all of that. So in it is the mesencephalic nucleus of 5. In it is the locus ceruleus. In it was the trochlear nucleus. In it is the dorsal tegmental. And then there are the nuclei are the raphe. This is the median raphe, but we'll come to that in a second. The raphe nuclei all share this that in common. All of the ones that bear the word raphe are serotonergic neurons. So that's a useful idea. Today we want in the midbrain, we want to talk about the dorsal raphe and median raphe. And to keep this simple, a couple of things. I'm not going to go over all this. What I would say is between the dorsal raphe and the median raphe, they project to nearly everything in the central nervous system. But the funny thing is, there's no overlap. They don't overlap where they're going. I don't think there's a single word that appears, a structure that appears in both of these subparagraphs. So the dorsal raphe has to do with two things, sleep and pain. And the median raphe for whatever it does, it participates in the limbic system, and this is LSD acts within the median raphe. All right. Both of the nuclei are primarily inhibitory, although on some occasions in certain sites they can be excitatory. But so let's just keep that. I think that's enough. These are very complicated talks with huge amounts of information. Dorsal raphe has to do with pain and sleep. Median raphe has to do with some psychogenic properties here. Who knows what its normal function is, and then inhibitory. And this was a good cartoon I found last night, I think, or the night before, and just showing you some of the projections of the median and dorsal raphe. So this is useful, and lots of notations also as to what it does. So a good cartoon. Here are the raphe. I think this is excellent. You can see the size of the neurons, the shape of the neurons, fairly extensive dendrites, medium-sized neurons. This is the dorsal raphe. Here, smaller neurons are the median raphe. Here's your aqueduct. This is the area of this was the dorsal tegmental. This is the fourth, all of that. And then here you have the trochlear nucleus. Sorry, this is locus ceruleus up here. Here on this view, which is a slightly different level, you have the trochlear nucleus. And here you have the dorsal and median raphe nuclei in this. So pretty good slides to help. Now the periaqueductal gray in the midbrain is heterogeneous. Again, that's what I said at the beginning. These are so hard because they do so many different things. They're so compact. And it's not to say that they do one, but they participate in all of these functions. And this is where, I've said this before, I'll say it again, it helps to think of this as a primitive nervous system in its own right, which is integrating all these other features which are coming in the worm, in the frog. I'll show you, the frog is funny, but in all of these more, in the earthworm is really, I just think it's a great analogy for what we do. We talked about axolotl and the primitive mammalian nervous system. So you see all of these different things. They're sort of related, they're not really related. The analgesia, vocalization, reproduction, aggressive behavior. I get a sense there's something common about them, but if you asked me what it was, I wouldn't really know. And then you have afferents to the periaqueductal gray that if there was one thing that all of this group had in common, it is that they participate in escape, again, into the primitive nervous system. We'll come back to this later when we talk about handedness, strangely. But in order to survive as a primitive nervous system, you have to eat and not be eaten. Okay? You've got to accomplish both of those things. So you have to be able to find your food and pick it out, but you also have to not be eaten. Okay? Not get burned, not find a toxin in your environment and get the heck out of there as quickly as you can. And if you inject morphine into the periaqueductal gray, you have profound analgesia, but profound analgesia. This is an old slide I've been using. I love this. It's just, we're parasagittal in the midbrain, and you can see the shape longitudinally and sagittally here of the periaqueductal gray. So it's not just a blob, and you can see the raphe nuclei. So this is really a nice slide. And this would be the aqueduct. This also, I found this one last night, and I like this one a lot. This is the concept of the periaqueductal gray. These letters are simple: dorsal, medial, dorsal, lateral, lateral, ventral, lateral. And this side are the ascending influences and projections, and this side are the descending influences and projections. So if you think of this as a primitive nervous system which is being accessed, it will help you keep this all straight. You're going in and out and in and out, okay? That's the way to think about it. The periaqueductal gray has strong connections to the limbic system, as any primitive survival nervous system better. If it can't relate to the limbic system, it's probably not going to survive very well, all right? Reproductive behavior, right? Things like that, avoidance behavior, anger, fight, flight, la, 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 la. You've got to have these connections. So the periaqueductal gray receives information from the habenula and the mammillary bodies. It projects to the medial forebrain bundle. But then this was interesting. It has some populations within the periaqueductal gray whose function seems to be a landmark and directional navigation. Again, it fits with the concept of finding your way around if you're a primitive nervous system. But it's interesting that you can demonstrate this in higher, in mammals. And so that I think was interesting. Also here in the midbrain, you have the interpeduncular nucleus, a very small nucleus in humans, mainly relating to the limbic system. Both of these are cholinergic. And here you have it. Here's the interpeduncular nucleus, right where it belongs. All right. And here's the dorsal tegmental nucleus. And now let's get up into the rostral midbrain. We have a long distance to go today. So let's get up into the rostral midbrain. Now, the rostral midbrain shows you the superior caliculi, which aren't quite as ovoid as the inferior colliculus was. You have, in addition, the V-shaped oculomotor complex, which is going to be really important. That's all ventral to the periaqueductal gray. And then you have the red nucleus, which is in the rostral midbrain. In addition, in the rostral midbrain, you have all of these visual centers, which we're going to talk about. The superior caliculi, the visual pathways, the pretectal region, the posterior commissure, the oculomotor nuclei, and all the nuclei which are for pupillary light reflex and accommodation. So this is classic midbrain, right? We look at this is classic midbrain. So let's just look and go around the clock here. Here's a superior colliculus, all right? Here are the oculomotor nuclei sitting ventral to the periaqueductal gray, close to the midline. Already you can see the V-shaped appearance. Here's a little spinal thalamic tract still working its way up. Here's the medial lemniscus way out here. This here is the brachium of the inferior colliculus. So that's an efferent pathway. Here's the medial lemniscus. Here's the red nucleus. You can make it out over here. Good, there's a red nucleus and a lot of fibers from the cerebellar pathways. Here's the substantia nigra, all right, the compacta and pars reticulata. Here's the cerebri. Here's the ventral tegmental decussation. I can talk about that. I want you to look at this. These are the root fibers of 3. We have a patient right now who has a partial third as part of a Benedict syndrome. And I use the analogy that it's like the third, for all that it's a cohesive nerve when it exits, isn't very cohesive inside the midbrain. It's not like like the 6th, which travels, or the 7th, which travels nice and compact. It doesn't do that. It's like a river delta coming together. And so the third is quite distributed within the brainstem. You can see it on either side. This, of course, is the interpeduncular fossa. And the interpeduncular nucleus is sitting here. Here you have striatal fibers. Here's, again, the medial longitudinal fasciculus is still, and in fact, is sort of getting home, if you will, as it finally makes its way to the oculomotor nuclei. All right, the central tegmental tract, we talked about that. That's still going. It has to still go. It has to get up to the thalamus. All right, and here's a nucleus of Darchowitz, and clearly demarcated. Again, these nuclei are well demarcated. We'll talk about all of them. And here's a cartoon. If you couldn't see it before, this to help you, and you can go back and forth between this diagram and the last diagram. All right, let's get into it. The superior caliculi. They're not so much a relay center. They're really not. What they are is for maintaining a reflex for orienting the head, the eyes, and therefore obviously the ears in space. Okay, so they're going to be interconnected with all of these. You got to have head and neck, they have to have eye, and of course they also have auditory. They're going to have all of those connections in order to carry out this job of orienting in space. They're going to detect if they have a single thing that really seems to run, is that they are for detecting objects in motion. Now, this is really interesting because when we get up into the visual system, you're going to see in the higher cortical centers, vision separates into a spatial orientation and an object recognition. And the dorsal portion, which is going to interact with the somatosensory cortex up in the parietal lobe, that's going to be for localization, all right? Maybe outlines of objects, but not so much object recognition. It doesn't care about color, for instance. It's going to care much more about movement and connecting it so that if you want to hit a tennis ball or swat the damn mosquito, that you can do that, okay? And this, it has to have a lower system for dealing with. And then there's going to be another system that will have color information, higher spatial resolution. And this is going to go from the retina all the way up into the higher visual cortex. Those two systems will be maintained separate. And that includes down here, and the superior colliculus clearly belongs to this dorsal aspect of localization in space, tracking movements, error correction, right? We talked about the parabigeminal nucleus for error correction. That does a part, it feeds back. Where did it go? It went to superior colliculus. See, so that's the thing. You need to know when you're on target and when you're off. And most of these neurons respond only to moving objects. All right. Now, we're going to need a retina. We're going to need information which makes it possible for that to do that. So we'll look at that in a minute. And the function appears to be to coordinate, the superior colliculus function is to coordinate head and eye movements. They're important, not surprisingly, for saccades. The information coming in matches that very well. What you have different kinds of retinal ganglion cells, and we will consider the eye in detail, okay? Part of the nervous system, right? Or the central nervous system, the retina. The Y cells are wide field neurons. They're not, again, not going to be color specific, not high spatial resolution, but they are all center surround. And as you will see when we talk about this in detail, the center surround is ideal for making a line. So it's just a dot and then a surround, but that's perfect for making a line. And then you have neurons whose job it is to, they're simple neurons that are called complex. And their job is to detect a moving bar of a certain orientation in a certain direction. So it's going to get that specific, and the cortex is going to be responding that way. And that kind of following information is the kind of thing that's being done in the superior colliculus. It's going to keep you on target of a moving bar with a certain orientation in a certain direction. And different neurons in the superior colliculus are going to be responsible for following a different orientation, moving in a different direction. That's really quite amazing. So the input is going to be from the retina, obviously. It needs that visual information, but it also comes from, look at this, from the occipital cortex and the frontal eye fields. Bless their heart. So I can tell it to follow this object, just like we saw for the tongue. I can just do it automatically because I'm a frog waiting to get a fly, and I got to get it right. I'm going to have to coordinate, including my tongue, because, and I got to catch that fly. Or I want to follow a baseball's course, or I want to follow it that he hit a home run, or am I going to take a header on this one, whatever it is. All right. So all of that, and all of this, in addition, as we already said, must happen, is you're going to have a lot of afferents coming in from the auditory system. Processed information from the inferior colliculi, less processed coming from trapezoid and relay nuclei. It's also going to have input from pars reticulata, and of course, especially from the neck, it has to know what is going on in the neck, or it really can't function because it needs to know the orientation of your head in space, or those targets are off. And so this is a good diagram, and it shows you just the incoming information. And here's the lateral genicula. This is not about lateral genicula. This is about superior colliculus. Notice the superior colliculus does not interact very much with the lateral genicula. This is for identification and object and specifics and that sort of thing. This is not, but it has to tell the cortex what it's doing and hear from the cortex what it should be doing. It is a laminated structure. It has 7 layers. The superior colliculus has 7 layers, all right? And that's what made me go back to the frog, okay? Because I keep trying to point you guys to phylogeny, okay? There was a time, this is a complicated structure, this superior colliculus, I mean, think about it, a 7-layered structure for visual field oriented movement in a particular direction. You get a lot of mileage out of this. If you're an animal trying to catch food, okay, there are goldfish that do this too, and they can pick these things out, and they're quick. I mean, also, have you ever seen the spitting goldfish? Well, there's a spitting goldfish that literally, this goldfish can, it sees the fly on a low-hanging branch. It spits water and knocks the fly off into the water and then eats it. Very sophisticated structure. It doesn't have a cerebral hemisphere like we do. So how's it doing that? This is a lot. This is a lot of neurons in here that are well organized for any function that you want. But in this case, it is to generate saccades and head movement. It's the superior colliculus, but in a more primitive nervous system. So it's now specialized. But look at the frog. Again, I literally keep thinking about the frog when I think about the superior colliculus. Look at the superior colliculus. This green is all superior. It's like, oh my gosh, it's a third of the brain. Why not? You know, why not? When you have this much, you know, complexity, you can do a lot. I mean, a Commodore 64 only had 64K. There's a lot more than 64K going on in the superior colliculus, okay? And these are details about the orientation of the superior colliculus here. And again, you have a much simpler cortex. The superior colliculus is really doing most of the work for the frog. I mean, there is a forebrain, there is a telencephalon, but it's not quite as big as the superior colliculus. Okay?
But a frontal is not that important for that.
The frontal is less important, right? Because it has less decision making, because the fly goes by, it follows the fly, it saps it with its tongue and eats, okay? And it has to know when to get out of there too. That will be important. It has to know when to leave, okay? Or it will get eaten. Of course, it can have different defenses, such as speckled skin that can warn it that you don't want to eat me, I am quite poisonous. That's a different matter altogether. The efferents from the superior colliculi, you can sort of predict where they're going to go. You have tectothalamic to the pulvinar. The pulvinar has a lot to do with vision as well as with language and higher cortical function. That is a late coming thalamic structure. We will talk about it in a lot of detail. It will send information to the lateral geniculus. It's going to send it, however, to the pedunculopontine reticular formation, to the rostral interstitial nuclei of the MLF for subcortical control of eye movements. It's got to now access all of the things that do eye movements. It doesn't, however, access the oculomotor nuclei. The oculomotor nuclei are not accessed by sensory structures, by telencephalic, by dorsal structures. They're motor, they're basal. They're going to be accessed and activated by structures of the reticular formation. And there are many different structures in the reticular formation, but the incoming sensory information plays on the oculomotor nuclei through the reticular formation nuclei, okay? So you have tectopontine, tectobulbar. These are the pontine reticular formation, to the paratrigeminal nucleus, to the cuneiform nucleus, which we'll look at in a minute, okay? And even to the cerebellar vermis. You're gonna have tectoreticular, which then go to interstitial nucleus of Cajal. This will control the vertical eye movement. This will not access the oculomotor nuclei. This will access the oculomotor nuclei, the interstitial nucleus of Cajal, the nucleus of Dershowitz, okay? We think of it as consensual pupillary, but it's much more complicated than that. And we're gonna come back to that, to the tectobulbar area and others for the parasympathetics. So, okay, let's set this up. Let's look at the oculomotor nuclear complex. I mean, come on, third nerve, talk to me about third nerve. So the third nerve or general somatic motor and that columnar organization we talked about, these are true, right? 3, 4, 6, and 12 are true somatic motor. Yeah, though lateral cell column, I find this, myself, I can't remember this level of detail. You have dorsal projects, so there's multiple nuclei. The dorsal projects to the inferior, the intermediate to the inferior oblique, the ventral to the medial rectus. It's like, if you can remember that, bless your heart. I'd have to look it up if I wanted to use that, which I rarely do. It's third nerve clinically, but interestingly, at the basic level, the medial column goes to the superior colliculus, to the superior oblique. Now, I do use the central caudal nucleus. That's the levator palpebral. Let me help you remember this. Remember the trick of using dizipramine to overcome cerebral ptosis or apraxia? So I used dizipramine, 10 milligrams. Okay, it will work. The dopamine is a direct neurotransmitter in the central caudal nucleus of Warwick, is its full name. It is part of the third nerve complex in the midline and inferior midline. It opens the eyes. It's the first nucleus that's activated when you wake up. You open your eyes. Okay? That's the central caudal nucleus of Warwick, part of the third nerve complex. The afferents you then have. The afferents to the somatic neuron do not include the cortex or superior colliculus. Most of the information is coming from vestibular or from the interstitial nuclei, the reticular formation of the brainstem, of the midbrain in particular. Here's a residual of the frog from the perihypoglossal. Okay, this is I think this upper left diagram is excellent, okay? You have the columns are separated by color. You can see the fourth, you can see the central caudal nucleus of Warwick, and you can see the visceral nuclei. Now, visceral nuclei are visceral, but they're also organizational, and so the visceral is a little too simple for that. In the complex, in the oculomotor nuclear complex is Edinger-Westphal, which is dorsal portion. That was what we were just looking at up here. Is the Edinger-Westphal up here. And these are the uncrossed preganglionic parasympathetics to the ciliary ganglion, okay? So that's what they do. They're also going to project downward and let other people know what they're doing. The nucleus of Darkschwitz is a part of the posterior commissure. It's also known as the nucleus of the posterior commissure. You will see that name often. They're the same. It is ventromedial to the periaqueductal gray, ventromedial. We actually saw it a couple of minutes ago. It projects to the vestibular nuclei for the semicircular canals, and it is critical. The nucleus of Darkschwitz, which we think of as for consensual pupillary light, actually is critical in the control of vertical eye movements. That I kept going back and forth of this because I've always worked with this clinical association of consensual. But it turns out when you look at the afferents and efferents, that vertical eye movement really is what it's about. And it's necessary for smooth pursuit in a three-dimensional space. It's critical to that. And so this is the kind of nucleus which accesses the oculomotor nuclei, as well as the interstitial nucleus of Cajal, okay? These are integrative neurons. They're also projection neurons. They're involved in the coordination of head and eye movements. So they're going to receive information about vision. They're going to receive vestibular information. They're going to receive second-hand or third-hand auditory information. They're going to integrate this information, and then they're going to let lower structures know what to do so we know when to turn our head, and it's going to access the oculomotor nuclei in order to actually turn our eyes. Okay, so nucleus of Darkschwitz, nucleus, interstitial nucleus of Cajal, and very good diagram here showing you the Edinger-Westphal is here, the interstitial nucleus of Cajal is here, okay, and these are the oculomotor nuclei right there. Tightly packed group, and that's why clinically, most of the time, we see them all at once. All right. Now the pretectal area, is the principal midbrain center for the pupillary light reflex. So the pretectal area is pupillary light reflex. This is actually a series of nuclei. The fibers are coming from the optic tract, the lateral geniculate, from the cortex as well, and from the posterior thalamus. It projects broadly and bilaterally to the visceral nuclei, the oculomotor complex, that is the Edinger-Westphal for pupillary control. And this is, I think, a really good diagram. So here we are in the midbrain. This is the pretectal area. This is the pretectal nucleus. It projects not to the oculomotor or anything like that. It projects onto Edinger-Westphal, and then Edinger-Westphal projects down to the ciliary ganglion, which projects to the sphincter. Okay, that's the system for pupillary, and they're going to interact with one another, so there's going to be a commissure for them, and then they get up into Darkschwitz as well in order to keep them working and to generate that consensual pupillary light reflex. Nice picture here, all right? Here's the posterior commissure. Now, the posterior commissure is an important landmark because generally speaking, the posterior commissure separates the midbrain from the diencephalon. That's your primary landmark for when you're leaving the brain stem and heading up into diencephalon. All right. Here's some other important structures. Okay, this is the brachium of the superior colliculus. This is an afferent pathway. Here's the superior colliculus. This is the pretectal olivary nucleus out here. They are then diagrammed here, so you could see pieces of it. Here's the olivary pretectal region, the olivary pretectal nucleus here, and you can see the information it receives from the retina, okay, which also goes to the superior oblique, as well as its projections out into the ciliary ganglion. Good diagram. We said the posterior commissure marks the transition. All right. Remember, nucleus of Darkowitz was also called the nucleus of the posterior commissure. Dorsal, it is dorsal, closer to the fourth, or where the fourth would have been, closer to the superior colliculus. Dorsal to the periaqueductal gray. It is rostral above the superior colliculus, and it is at the transition from the aqueduct into the third ventricle. So this is all the way at the top now of the midbrain. It should be if it's a transition. You have fibers traveling in the posterior commissure for pupillary light, upward saccades, the interstitial nucleus of Cajal for vertical and integrative eye movement, mixing head and neck, nucleus of Darkowitz for both pupillary and upward saccades. So all of that going through the posterior commissure. And then also the levator information from lower central caudal nucleus. And lesions in the posterior commissure, not surprisingly, will impair consensual light, as well as lid movements and vertical eye movements. That's all in the posterior commissure. Now, as an aside here, given where we are localization-wise, I want to talk about the subcommissural organ. This is interesting. So we're leaving eye movements for a moment. All right. This subcommissural organ is interesting. It is a modified ependymal plate in the roof of the aqueduct. All right. It is right beneath the posterior commissure where it's located. It is for sensing CSF. So if you wanted to sense the CSF and you wanted to know what was going on in the CSF being made in the cerebral hemispheres, I guess if you want, you could put sensors all over the place. But what you really could do is let the CSF come to you. And where it funnels through the aqueduct, you could sample the whole thing all at once. So the subcommissural organ is a series of ciliary columnar cells with both secretory and absorptive function. And they are sensing what's going on in the CSF. Their mode of action is really interesting. We knew that when I did this 3 years ago, we knew that they made this molecule, this subcommissural SCO, subcommissural organ spondin, was the protein that was synthesized. People didn't understand quite very well what it was, but in the last 3 years, there's been quite a bit of advance. So SCO spondin is a large glycoprotein. It can exist within the CSF either as a monomer or a polymer. When it polymerizes, it makes long fibers, and these fibers extend all the way from the midbrain down into the spinal cord. There's a long way for these polymers to go, and they bind and regulate CSF molecules. So their job is they're a polyfunctional large protein. They can make large aggregates of themselves. Who knows what kind of quantum tunneling and things like that go on in a molecule fiber that's that long, a polymer. But they can also grab on to certain molecules and then they can sense what's going on, right? And how they execute is still maybe 3 years from now. Juan can give this lecture and we'll see what it's for. This is a subcommissural organ. Here's the aqueduct. Here's a periaqueductal gray. Here's a subcommissural organ. So very interesting little structure. Here it is in detail. Columnar cells, ciliated columnar cells. You can see all that. Very pretty picture. All right. Midbrain tegmentum. Not a lot, we said, of the midbrain. Now we've talked about so many things. It is the area of the midbrain that is ventral to the aqueduct. It is dorsal to the substantia nigra. I used to keep substantia nigra in this lecture. I took it out a week ago when I looked at the lecture. I thought, no way. I'll fit it in movement disorders
Yeah.
and leave it at that. The tegmentum of the midbrain contains the oculomotor complex, the mesencephalic reticular formation, and the red nuclei. We've dealt with all these others, so let's focus on the red nucleus. All right, a picture, crescent violet, aqueduct, tegmentum, right? So here's the dorsal tegmental nucleus that we've been talking about, the mesencephalic reticular formation. Here's the peripeduncular nucleus that we're going to talk about when we talk about the crus cerebri in a minute. All right, here's the red nucleus. This is what I wanted to focus on. You have small cells and larger cells, but you can see the size. Once you see it, it's actually a fairly big structure. Red nucleus is not small at all. Here were the oculomotor complex here. All right, what else do we got? That's largely it. Red nucleus is what I wanted to focus on. Here's how I think of the red nucleus, and you can think about it however you want. Here's how I think about it. If I ask you to pick up a chair and move it across the room, you're using your red nucleus to hold the chair. If I ask you to go write something on the board, you're using your red nucleus to hold your proximal arm fixed or move a little bit so that the rest of your arm can write and not be all over the place. The red nucleus is stabilizing the proximal arm and shoulder together. That's what the red nucleus does, as far as I could tell. And it's set up for that. And it's more strictly related to flexion, but I think if you just think about the act of picking up a chair and carrying it, right there, as you literally imagine picking it up, you can feel the red nucleus hold your arms in place, okay? Again, if you go to write on the whiteboard, that's what it's doing. That's red nucleus. That's when you use your red nucleus or any other activity like that. If you say, what about a bow and arrow? I go, absolutely, why not? Right, bow and arrow, sure. How about swimming? Yeah, why not? Anything that involves a coordination in a relatively fixed position of the proximal arms, think about the red nucleus playing a major role. The phylogeny, as well as the ontogeny, that is development within the developing nervous system, both of those, the red nucleus is associated with the dentate. So that's the association here. The dentate grows in size and importance as we move up in phylogeny. It appears later in ontogeny. It grows later in ontogeny. As it's growing, so is the red nucleus growing, either phylogenetically or in the development of the fetus. Either way, they grow together, okay? So they're clearly strongly related to one another. The afferents to the red nucleus are the ipsilateral premotor cortex, so it knows what it's doing, premotor cortex, not the corticospinal tract. Premotor cortex. It has to be involved in planning. If I'm going to pick up the chair, if I'm going to write on the board, yeah, okay, I have to hold still, but I have to hold still within the context of reaching higher on the board, reaching lower on the board, that kind of stuff. You also have afferents from the contralateral cerebellar cortex through the dentate, as well as through the emboliform and column. The efferents are back to the cerebellum, rubro-bulbar, back to the cerebellum, also the cranial nerve 7 and cranial nerve motor portion of 5, and the rubrospinal tract down especially to the cervical spinal cord as you fix your upper body in order to do this. It works if you think about it that way. Here's a great picture of the red nuclei. All right. So often we're trying to see the red nuclei in the cerebellar decussation, and they get lost by all the fibers of the superior cerebellar peduncle. As they're moving toward the decussation, they are infiltrating the red nucleus. But when you move up to the most rostral portion, notice the posterior column, to the most rostral portion, so you really can see the red nuclei here, which are surrounded by fibers, but not obscured by them. And this was a nice cartoon I found last night, sort of showing you ups and downs. It's premotor coming into red nucleus, going down to the spine, also information coming in from the cerebellum, also coming in from the pretectum. All right, very nice. Good. The reticular formation, in the midbrain, when we talk about the reticular formation, the pedunculopontine nucleus is a very important part of it. It's ventral to the anterior colliculus, You have input from the cortex, from the medial pallidus, from post reticulata. These are related to one another. We'll come back to that another day. You have efferent from the reticular formation going to pars compacta. This portion has a lot to do with arousal, attention, locomotor, that sort of thing. You have the cuneiform and subcuneiform portions, nuclei in the reticular formation. They are mostly for pain and the elaborate defense behaviors of to avoidance of pain. And this is when the cat puts on its display, ears back, hissing, showing its teeth, that sort of thing. That sort of thing. It's using its nucleus cuneiform and subcuneiform nuclei, right? Pupillary dilatation and all that, all those things. So mesencephalon, right? Here's the pedunculopontine tegmental nucleus.
The fact that they are called uniform is just a matter of shape, or is a matter of redundance with the cuneus and precuneus officers?
I think the former, Han. So their shape primarily.
Okay.
Yeah. Now, yeah, so you can sort of see that way. There are some more pictures. Here's a cuneiform. This is the yellow is the nucleus cuneiformis and subcuneiformis, and the green is the pedunculopontine tegmental nucleus. And I think this, when you look at the reticular formation, yeah, it's like, oh, so when I keep saying this is a lot, this is enough nervous system to run an organism, I'm serious. Okay, this is a lot of nervous system. When you start thinking about it from the computing point of view, functional point, multifunctional, integrative, you got a lot of resources here. We don't, and I think it's extremely important in the development of the nervous system, especially phylogenetically, to realize that we don't throw any of it out. We keep it all, and we use all of it, and we just start adding more layers and more layers, more control, more sophisticated, more integration, okay? That's what's happening. You don't get rid of any of this. It all remains useful. And then I think we're gonna do cross survey. We're just about done. The reticular activating system is this neuronal network. It's central core, it receives sensory information, but it doesn't use it as sensory information. It's not modality specific. It's any sound, touch, pain, vision, any of those can activate the reticular activating system, which simply alerts the rest of the nervous system. That's what it's doing. Primarily, it's going to ascend up to the interlaminar nuclei of the thalamus. And there's a lot of discussion about consciousness. There are a lot of ways. There's a physiologic approach to consciousness, which is having some progress right now. There's a certain wave which only appears when the person is conscious of a behavioral event. And that wave goes well. We don't really know where that wave comes from. It might come from the interlaminar nuclei. Did a search last night seeing if we had made progress on that. The answer is no. But this is arousal. There's arousal consciousness. Consciousness is a very complicated complex. But I'll tell you this, you're not going to be conscious if you're not aroused.
Right.
Okay, that much I'm certain of. Okay, so it clearly plays an important role. And if you knock out the midbrain tegmentum, we all know this from the ICU. If you knock this out, there's no arousal. There's no arousal of the patient. There's no arousal of the EEG. So that's for certain. And then we're going to finish with just a couple slides here on the crus cerebri. The crus cerebri is where all those descending fibers are coming out of the cerebral hemisphere. We've talked about this, but I just want to add one or two quick things in terms of somatotopic organization of the crus cerebri. Obviously, they're the most ventral portion. That's what makes the midbrain look like the midbrain. You have the medial 3/5. It's the cortical spinal and cortical bulbar fibers. All right. Lateral goes to the leg, medial goes to the face. The extreme fifth of the crus cerebri is frontopontine. The extreme lateral fifth is corticopontine from the rest of the cerebral hemispheres. So both medial and lateral thirds, practically, of the crus cerebri, all right, are cerebral, okay? And that medial is the sort of the information that we deal with most often clinically in terms of corticobulbar, corticospinal. There's a separate little group called pes lemnisci, the foot of the lemniscus, which is separated and has to do with reticular formation. And we've already talked about this 1 out of 20. And here you are. So here's a picture of a Weigert stain, not a Weigert stain, but a picture of a Weigert stain. And here you have the organization. We talked about this pes lemnisci going to the reticular formation, frontopontine, corticobulbar, corticospinal, and then the rest of the cerebral hemisphere. Okay, and that's it. All right. I almost changed this last night to say, don't make me activate my nucleus cuneiformis. I left the old one for tradition, but I must say I was tempted. All right, that's it, guys. See you next week. We have clinical correlations coming up on the brainstem. That'll be a lot of fun. We'll help solidify all of this detailed anatomy, and then we will stay in the posterior fossa, but clearly change gears as we begin to talk about one of the consummate pieces of the universe, and that is the cerebellum. Okay, we'll get to that very shortly. Bye, everyone.